Thanks a lot for the invitation and to all of you for coming. Okay, so as Alain said, I will present you the work we've been doing in the last 4 or 5 years, at least, on the development and more recently on the evolution of brain commissures. So what are brain commissures? So most of the animal species, they are bilaterian, so they have a right side, a left side, a front and a rear. And they also have neurons in the brain that sends axons across the midline to connect to neurons on the other side of the midline. And most of them, they cross the midline only once. I will show you at the end. There are a few exceptions, but they're very rare, where an axon crosses twice the midline. And again, you can see in all those species, you can see axons that can either, when they cross, be together, forming a defined tract called commissures. So we also have them, you will see later, in the mammalian brain. Or most often in the hindbrain, for instance, like here in the mouse, those axons, they cross everywhere along the axis. So it's not like you have a particular structure where they can cross. It seems that they cross everywhere. I wanted to show you this. So it's unfortunate that Christine is not here, but most of this was done by Christine or by Carl Mason, which is that it's clear that in evolution, during evolution, there have been changes that have affected the commissures and the way they are developed, and also the proportion of axons that cross or don't cross the midline. And work by Christine and others have clearly shown that in a vertebrate brain, the proportion of axons that cross differs between species. So for instance, if you take fish and birds or premetamorphic frogs, all the axons from the left eye will go to the right side, and all the axons from the right eye would go to the left side. Now, if you move up, if you want, if I can say, in evolution, if you go to mammals, then there's a contingent of axons now that will project ipsilaterally, and the choice will be made at a region called the optic chiasm, and that's where the axon will decide to either cross or to stay ipsilateral. And this proportion of ipsilateral axons is increasing again, such as in mammals. And in humans, it's about 50%. In the mouse, it's about 3-4% that stays ipsilateral, but 97% will cross. And what Christine has found is that in the frog, for instance, as I said, initially before metamorphosis, everything is crossing. But after metamorphosis, there's a proportion of axons that goes ipsilateral. And she shows that it's linked to the expression that appears during metamorphosis of a repulsive molecule called ephrin B that now becomes expressed at the chiasm. So, and then the axons that will become ipsilateral, they will be repelled from, and will stay on this side by ephrin Bs. And there's a correlation between the amount of ipsilateral axons and the amount of binocular vision that you can have. So one thing here that it does, the fact that now you have ipsilateral connections in the system, is that it also allows you to have depth perception. So depth perception is really, the appearance of this is really due to the appearance or change in the proportion of commissures. So one of the most famous commissures is also the corpus callosum, that is known for a long time, even before people knew there were neurons in the brain. And it's a structure that connects the two hemispheres in the brain. And they were studied, as you can see, for a long time. And again, it's something that differs in evolution. And it appears in mammals, but you don't have it in monotremes. And then it comes in marsupial, and then the proportion of axons increases again and is higher in human, in mammals. So again, you can correlate the amount of commissures in the brain with the evolution. So another interesting feature about the function of those commissures is that this ratio and this ability to project to one side or the other can have some effect if it's perturbed during development. And I'm just showing you one example here of a tract, which is called the corticospinal tract, that connects the motor cortex in human to the motor neurons, it's direct in human, on the opposite side of the spinal cord. So those axons, they leave the cortex, they grow along the brain, and when they reach the border of the spinal cord, then they decide to cross, and they will project mostly to the contralateral side. And there are a few diseases in humans where this choice is not normal, and some of the axons, when they reach the midline, they will also project to the other side. And the consequence for people is that they have so-called mirror movement. So it means that when they send some motor instructions, then the two, for instance, when they want to move their finger on this hand, then the fingers on the other hand will also move at the same time. So that's why they are called mirror movement patients. And there are a few mutations that were identified, in particular here in Paris by the group of Emmanuel Rose and Alexis Brice, in a receptor for netrin, I'll talk about that later, DCC, and in the DNA repair gene, RAT51. So a few examples like this. So for more than 25 years now, there have been many groups that have been working on the mechanisms that control choice decision at the midline. And this is just a diagram that summarizes some of the findings about the molecules that are playing a role in this choice made by axons, either in the spinal cord or hindbrain, or in the forebrain, in the corpus callosum. You don't have to remember everything. I will come back to some of them later. And as you can see, it's quite complex. There are changes. One of the questions that is asked here is what happens when an axon is first attracted to the midline, why it doesn't stay there, why then the axon will cross and move away. And it's clear that there are changes in the receptor expression, there are changes in the appearance of new receptors and other things, but I won't talk so much about that today. So just keep in mind that it's complex, but there are some similarities in the forebrain and in the hindbrain about some of the molecules that are shared for all the commercial system. So for today's talk, I will mostly present the work we've been doing on roboreceptors. So roboreceptors are found in all animal species, and you have them in C. elegans, you have only one roboreceptor, but in most other species, you have more than one roboreceptors. And it's clear, so if you start from the end here, so in mammals and in humans, for instance, you have 4 robo genes called robo1, robo2, robo3, and robo4. And they are on 2 different chromosomes, and they are head-to-head with absolutely no gene between them. And it's clear that in evolution, they have been produced by tandem duplication of the ancestral gene, and then there have been 2 subsecutive duplication of this genome, and then some also losses. And there are 2 possibilities. We don't know which is which. But the end result is that from 1, you go to 4 receptors. And you can take any kind of vertebrate species. It's always the case. You always have those 4 genes on 2 different chromosomes head to head. So the slits, they are the ligands for the roboreceptors. And as I will show you later, there are some interesting features about how they bind the receptors. Here is just to show you what happens when you apply slit ligand on a retinal ganglion cell growth cone. You can see that it will induce the collapse of the retinal ganglion cell growth cone, and that is mediated by roboreceptors. So slits are diffusible protein, and they can induce a growth cone collapse, and they are very potent repellent in most systems. They not only control the growth of axons, they are also controlling cell migration and cell-cell interaction in most developing organs. So this is just a structure of the roboreceptors. It's an immunoglobulin superfamily member. It has a 5 Ig domain, 3 fibronectin domain, and then various domains in the cytoplasmic tail, which are called a cytoplasmic conserved domain. So CC0, CC1, C2, CC3, and I'll come back a little bit to this later. And you can see from this diagram here that there are some differences between the various roboreceptors and also between receptors between species, but this was not studied very extensively so far. And again, I will discuss that a lot today. Okay, so what happens when you play with the roboreceptors? So I just want to show you this, which is, I think, one of the nicest examples of a work that was done by Chi-Bin Chen, where he injected a tracer in one eye, and then he can follow the growth of the axons from the eye to the chiasm, and he can show what happens when they reach the chiasm. And here, in this case, you have a mutant called Astray, where Robo2 was mutated. And as you can see here, if you just remove receptor, actually from the eye, when they reach the chiasm, the growth cone here becomes disoriented. It doesn't know where to go. Some axons will cross, some of them will go back, they will go to the other eye. So here, just removing receptor, the growth of the axon to the chiasm is okay, but then the choice of the axon is completely lost. So I think it really shows you that those roboreceptors are really important for guidance. So today I will just talk extensively about another roboreceptors, which is Robo3. And Robo3 is interesting because in the hindbrain and spinal cord, it's expressed by all commissures. So one way to show you this is to show a whole-mount view of a hindbrain of a mouse embryo that was stained with Robo3. So we see the spinal cord, and we're moving up to the hindbrain. You can see the cell bodies of the commissure neuron, and you can see the axons crossing the midline. So you can see all of them, all the axons in the spinal cord that are crossing, they express this Robo3 receptor. So ROBO3 is present in, ROBO3 is present in all vertebrate species, and it has, as you can see here, it has, again, the structure of a ROBO-receptor, 5 Ig domain, and there are, the highest homology, it's at the level of the first 2 Ig domain, Ig1 and Ig2, but there are some differences. There are also different splice variants of ROBO3 that differs either in the N terminus, that are called A and B, and or that differ in the cytoplasmic tail, and I'll come back to that later. So here, I'm just showing you some immunostaining with an antibody against ROBO3 on mouse spinal cord, chick spinal cord, xenopus hindbrain. And in green, you can see that ROBO3 is expressed by commissure neurons. And in fact, at least in the mouse, ROBO3 is and in mammals, ROBO3 is expressed by commissure neurons just when they grow to the midline, which is called the floor plate in vertebrate. And after they have crossed the midline, ROBO3 expression is downregulated. So all commissures express it in the hindbrain spinal cord, but then it's downregulated. I just want to mention here, and I might come back to that later, that it's not expressed in the forebrain in the corpus callosum, for instance. It's just in mammals in the spinal cord and hindbrain, except one structure that I will come back to at the end. Okay. So what is interesting as well with ROBO3, and that's also why we are working on it, is that there's a rare disease in humans called horizontal gaze palsy with progressive scoliosis, so a complex name. That means basically that the patient that suffers from the disease, they cannot move their eyes laterally. Okay, they can move their eyes up and down, but if you ask them to look on the side, they will turn their head, they cannot move their eyes together. Okay? And what they also have is this very severe scoliosis. So it's autosomal recessive mutations, and you have about 20 families that were identified so far, and they all carry mutation in different amino acids along the sequence of the ROBO3 gene. And what is interesting, and what was found 10 years ago, is that in ROBO3 patients, the corticospinal tract is now ipsilateral. So it means that in the patients, all axons for the motor cortex on the right side will project now to the right side of the spinal cord. And what is also interesting is that all the ascending projection, all the ascending sensory information from the spinal cord to the cortex that also normally cross to the other side, they will also be ipsilateral. And in fact, in all the systems that have been looked so far, although the patients, they survive well, so no one has looked at their brain post-mortem so far, but for everything that was tested so far, it seemed that they lack commissures in the spinal cord and the hindbrain, but they still have a corpus callosum. And what is surprising is that despite lacking all those commissures, so most of the commissures in the brain are here, although they lack it, those patients don't have mirror movement, they are coordinated, they have no mental retardation, and as I said, the only feature that they have is this horizontal gaze palsy and this scoliosis. So I won't show you this today, but we made a mouse model of the disease, and we could show that this defect in the eye is due to an absence of connections between neurons that control eye movement and coordinate the right side and the left side. So when you move with both eyes, you have to contract muscles on this side, to release muscle on the other side, and this is due to a commissure projection that is gone in those patients. Okay. Okay, so this is what is known so far and proposed so far for the mechanism of actions of ROBO3. And that's the working model that people have been working on for several years now. And I just made a simplistic view of the commissure guidance at the midline, where you have one main attractant called netrin-1 that is binding to a receptor called DCC, and this will attract the axons toward the midline. Then you have the slit repellent, 3 slits in vertebrates, slit 1, 2, and 3, and they bind to those ROBO-receptors, and when they bind to receptors, there is repulsion. So ROBO3 is expressed just during this phase, before axon cross, and after they cross, it's downregulated. And the idea is that if you add repulsion to the beginning, then axons wouldn't be able to make it the spinal cord because they will be repelled by slit. So you need to have a mechanism that prevents this repulsion initially, and it was proposed that ROBO3 plays this role by, when it's present, inhibiting this repulsion, thereby allowing the attraction. So you have ROBO3, ROBO1 and 2 cannot trigger repulsion, axons are attracted, and after crossing, ROBO3 is downregulated, and now you can induce repulsion, and ROBO as well will bind to DCC and block the attraction. Okay, and there are two, what is even more complex, and I won't go too much into the details later, it's just to mention, for some of you who know about this, you have two isoforms of ROBO3. One, ROBO3.1, seems to be expressed before crossing, and after crossing, for a little while, there's a second isoform called ROBO3.2 that is expressed. And the second isoform, it's induced by you keep one intron here, 26. So in the Robo 3.1 isoform, you go directly to exon 26 to exon 27. In this Robo 3.2 isoform, you go to the intron, and then you have a stop codon, and that produces a shorter protein. So there are reports that have shown that Robo 3.2 is important for axons to use the spinal cord. Just to point you one feature of Robo 3.2, that's something we're starting to do, that shows you that although the Robo 3.1 isoform is very well conserved in all species, now if you take the Robo 3.2 isoform, you can see in different species there are no conservation. So this is a bit surprising. You have something that's supposed to be important, a splicing that's supposed to be important for function or to change the function or the guidance. But then it means that if Robo 3.2 has a function, the function, or at least the tail, is different in all species. So you could think that maybe this switch of 3.1 to 3.2 is maybe to inactivate with receptor, and that maybe Robo 3.2 by itself doesn't really have a function. But it's really something that is worth studying. But again, you can see that here in all species, you have different isoform of Robo 3 if it exists. Okay, so I mentioned this Robo 3, and we were a bit we wanted to go into the structure and study a bit more precisely, for reasons you will understand later, what could be the mechanism of action of Robo 3. What I show you on the previous slide, it doesn't tell you that slit binding to Robo 3 trigger any kind of interaction with Robo 1 and Robo 2. So the structure of the binding domain of Robo or slit to Robo has been solved, and it's known that it's a domain called the slit2D2 domain that of Robo, of slit, that is binding to the first IG domain of Robo1 receptor. So that's where it binds. The structure is shown here. And then the structures has allowed a researcher to identify some amino acids that are linked, that are located in the regions where slit binds to Robo. And I've been showing you here some of them. And it's a work that was done by Erhard Oenester in London. And when we looked at this sequence and align it and compare it to Robo 3 sequence, we're a bit surprised, because so this is just a small stretch of the IG1 domain, and this is Robo 1 and Robo 2, and here you have different Robo 3 species. And you can see that we found by aligning it that some amino acids, like this asparagine here or this lysine here, it seems that they are not conserved in the Robo 3 in mammalian species. So they are replaced by a proline or an arginine. And here you can see again, this leucine here is a proline when you look in mammals. In all the species, it's never the case. And this is not only in vertebrates. You will see later, if you go to C. elegans, to Drosophila, to all species, it's always the same thing, NKL, it's never this. And based on the structure of those amino acids here, in particular those two, the proline here, sorry, this asparagine here, and this leucine here, they are supposed to be very important and key for slit binding. So it was suggested that maybe, somehow, Robo 3 mammals doesn't bind slits. So I'm just showing you here more of the alignment. Again, Robo 1, Robo 2, you can see it's always the same in most species. And in mammals, it's never the case. You always have two proline here. So that was suggesting that maybe Robo 3 in mammals doesn't bind, is not able to bind slit. So what we did, here we express in cells Robo 1, Robo 2, and we apply to the cells some fusion protein of the binding domain of slit2, slit2D2, fused to the alkyne phosphatase enzyme, and then when you do the binding afterwards, you can reveal the activity of the binding by revealing the alkyne phosphatase, and it's a very sensitive assay. So here you can see Robo 1 binding, Robo 2 binding. Now, if you mutate this amino acid and turn it to a proline, you still have binding. But if you mutate this leucine to a proline, you lose completely the binding. So one of those amino acids is sufficient to suppress the binding. Again, now if you take Robo 3A1, you don't have any binding. And we tried that several times, and there's been no binding of slit to Robo 3A1, or no strong binding for sure. And what was surprising, now if you change this proline to the leucine, you can restore some binding. So we studied that more extensively. And in fact, what we found is, as expected of the structure, slit can bind to zebrafish Robo 3, it can bind to xenopus Robo 3, it can bind to chick Robo 3, but it doesn't bind to the mouse Robo 3. And here, if you just mutate again this amino acid, this leucine to a proline, again you lose completely the binding when it's zebrafish. And in ZPs as well, if you mutate those conserved amino acid, you can also use SLIT binding, and we made some structured analysis. So it's really that those amino acids, they are key for SLIT binding. As expected, if you mutate them in a roboreceptor that binds SLIT, you lose the binding. But if you restore them in a mammalian Robo3, then you gain binding. Okay? So SLIT doesn't bind to Robo3 in mammals. And then we went to discuss with Hugues Ruscroyus at the École Supérieure, who works on evolution and gene evolution, to try to see how far we could go and if those results could be extended and were meaningful in terms of evolution. And what Hugues and Amélie in his lab and François in my lab did was to compare more extensively the sequences of the Robo genes, either Robo1, Robo2, and Robo3, and using some technique and software, such as this panel software, where you compare genes, so you enter all the sequence, and then the software will tell you whether there are some amino acids that could have been subjected to positive selection. So doing evolution is very selective pressure on some amino acid that have changed the property of the receptor and make it in a way maybe better and favor the selection of one amino acid. So if you do that on the whole sequence, you can see that in Robo1 there's hardly anything, one maybe here. In Robo2 there are no selection, but if you take Robo3, you can see there are many amino acids that are selected. And in particular, in the branch that goes to mammal, we found that 11 amino acids have been most likely positively selected. And I'm just showing you here where those amino acids are. You can see there are two, I'll come back to that in the IG1 domain, which is again the binding sites. And you can see you have many in those fibronectins here, and just a little bit in the cytoplasmic domain. So again, it's something that suggests that Robo3, when you went to mammals, things happen that have changed its structure and possibly its function. And we focus and I will focus mostly for now on those two amino acids here. So again, if you align it, this is where they are. And if you do just the panel on the IG1 domain, you can see there are absolutely no selection for Robo1 and Robo2 in the IG1 domain in all species. But again, you have selections when you move to mammals for this IG1 domain. And the two amino acids that are selected are, again, this proline that was also found to be important for SLIT binding. And here, an alanine residue that is a serine in all other species. And so far, what we've done is to mutate this asparagine here, which is the one that you have in Robo1 or in Robo3. So we take here the zebrafish Robo3, we mutate this N to a P, and you can see that this is enough to suppress SLIT binding. So this amino acid, again, that is conserved, that is not conserved, that is different between non-mammalian and mammalian Robo3. You mutate a single amino acid that is positively selected, and you lose SLIT binding to Robo3. So it shows that this type of analysis is very interesting, and something we are planning now to do on all axon guidance receptors to try to see if there's something similar in other species. But again, it shows you that most likely during evolution, this has had some consequence on gene expression and on gene function. Okay, so what we found also in the lab, so that the work of Eike Blockus mostly, is that there are also differences in the way the receptor responds to SLIT. So not surprisingly, if you express Robo3 and you apply SLIT, you don't get any change. We just look at here the phosphorylation of the cytoplasmic domain of Robo3. Here you don't get anything, but there's no binding. Now, if you do it on the zebrafish Robo3, you can see that it's highly phosphorylated in basal level. And when you apply SLIT, you have a significant decrease of Robo3 phosphorylation. And now it is interesting, if you restore SLIT binding to Robo3 by mutating the mouse Robo3, so this one is able to bind SLIT, you can restore, you can see that the phosphorylation is higher, and now Robo3 can be dephosphorylated when you apply SLIT. And in the opposite, now if you take zebrafish that is mutated, you don't have any more SLIT binding, and when you apply SLIT, you don't do anything to the phosphorylation is lower. So it really shows you just by changing the amino acid, you can restore SLIT binding, and you can also restore some kind of phosphorylation. So to summarize here, in non-mammals, the receptor can bind SLIT. When it's phosphorylated in basal condition, when SLIT is bound, then you have dephosphorylation. But in mammalian Robo3, you can apply SLIT, nothing happens. So it means that Robo3 function must be different. Okay, so what is its mechanism of action? So to understand this a bit better, I will need to present you the system we've been working on for many years now, which are some pre-cerebellar neurons. It means some neurons in the brain, in the hindbrain, that send axons to the cerebellum. That's why they're called pre-cerebellar. And they all come from a dorsal during development from a region of the brain called the rhombic clip, that is dorsal. And all those neurons that are born dorsally, they migrate to the ventral side of the brain toward this midline called the floor plate. So you can see here it's the work of Pavazelina. We have been injecting a DNA encoding GFP, and then he has age-operated, and the DNA will be incorporated in the rhombic clip. And if you do it late enough, you will just target the GFP expression to the pontine neuron, which are those neurons here. So that migrate rostrally here to the brain. And you can see that you have the rhombic clip here. It's a flat molecule of the hindbrain. You can see all those neurons that are born here. They will form a compact tract. They will migrate across the brain. And then they reach a turning point. And they will migrate then ventrally toward the brain. The cell body will stop here. And the axon will cross and go to the cerebellum. And you can see a close-up here of some migrating pontine neuron. They have this unipolar morphology, a single leading growth cone, and all those axons and growth cone, they turn toward the midline. Okay? So as I said, if you make a section now at the level of the pons, they go to the midline. 95% stop at the midline. The axon will cross only. And they express various markers that allows you to recognize them in the embryo, such as PAC6 or another transcript factor bar HL1. And obviously, they express very highly ROBO3 receptor. So if you incorporate GFP in a wild type, you can see the rhombic clip axons from this stream. They migrate, they reach the root of the trigeminal nerve, and then they turn ventrally, and then they migrate toward the midline. So now if you remove ROBO3, you incorporate in the mutant, you can see that the axons, they leave the rhombic clip, they migrate to the trigeminal nerve, then they go ventral, but then suddenly they cannot make it closer to the midline, and they turn dorsally. So you remove ROBO3, they are not able to make it to the midline, and instead the distance is about 500 micrometer away from the midline. So you can see the difference when you do an overlay between two different embryos. This is a mutant. Everything is normal at the beginning, even when they start migrating, but then there's a point where they cannot make it anymore, and they have to grow dorsally, and their axon will project to the cerebellum, but on the wrong side. And this is what you see in human. In human patient, the pons is also dorsal. The axon doesn't seem to make it to the midline. So that's why the full knockout. We also have a conditional knockout. And if we cross this conditional knockout with 3 locks to a Cre line where Cre is expressed not only there, but is expressed highly in the pontine progenitors, you can see that you can mimic the phenotype. Now all the neurons will also migrate dorsal. So just remove both in the system is enough to make them turn dorsal. So the first thing we tried was, okay, you have this phenotype here that is it's 100% of the case, it's fully penetrant. What would happen if we re-expressed ROBO3 in those neurons? Can we make those axons, those neurons be able again to turn ventral? And what happens if we do it to a mammalian form or a non-mammalian form? Is the result the same? And the result is not the same. So here this is what we get when we overexpress. So there's GFP to see the operating neurons. And we could also see that the re-expressed ROBO3, I don't show it here to save a little bit of time. You can see that the neurons, some of the neurons that express GFP will be able not to re-migrate towards the midline. But if you express the zebrafish ROBO3, then the axon and the neurons, they will still stay dorsal. So again, it shows that just a mammalian form can rescue the ventral migration, but a non-mammalian form cannot rescue the ventral migration. So the next step was to study whether, okay, it doesn't bind slit. Here the migration, ventral migration is prevented. Could it be that ROBO3 mediates attraction and has nothing to do with repulsion? So again, the strategy is the following. We electroporate GFP in the embryo. It will only go in the pontine neuron when they migrate, so you will see them because they're green. Then we can make a dissection of the rhombic clip. We can put it in a collagen gel and place floor plate, and you can see the neurons, they leave the explant, and they migrate, they are attracted by the floor plate. If you put netrin expressing cells, it's the same thing. They form streams of neurons, and they migrate toward netrin or towards floor plate. So it's a very robust assay. And the next step was to do the same thing by age-operating some ROBO3 mutants, dissecting the rhombic clip and culture next to floor plate or netrin. And if you do this, you can see that you are completely losing the attraction. You can make movies of those explants, and you will see that the neurons are migrating inside the explant. So it's not like they are not able to migrate, and in vivo they do migrate. Okay? It's just that they are not attracted anymore by floor plate. Now, if you take netrin expressing cells, the cells are here, you don't see them. They are, again, you can attract wild type, but then if you take mutant, you don't attract anything. So removing ROBO3 is sufficient to prevent floor plate attraction and to prevent netrin-1 attraction of pontine neuron. And more recently, we've been working with Marc Tessier-Lavigne, and he found that in the spinal cord as well, there's a very strong and significant reduction of netrin attraction when you take spinal cord explants from a ROBO3 mutant. So it's not something that is only in the hindbrain. It's also true for spinal cord, although in the hindbrain it seems to be fully required, and in spinal cord it's not completely the case. Okay, now if you look at the netrin mutant, so there's some netrin knockout as well, so we remove, which is this molecule at the floor plate, you can see that the phenotype here is again very similar to what you see in the ROBO3 mutants. Neurons leave the rhombic clip, they migrate, they reach this root, they start going ventral, but then they stay dorsal. If this is just a root of the trigeminal, that's where they make the choice to turn ventrally. So the phenotype is very similar to the ROBO3 mutant. So we said, okay, maybe the simple thing is that netrin binds to ROBO3, so just at the binding. So we made some fusion protein of netrin fused to alkaline phosphatase, and we binding to cells expressing ROBO3. And in fact, it doesn't bind to ROBO3. It doesn't bind to chick ROBO3, to zebrafish, to mouse. It doesn't bind to any of the ROBO3 receptor. And it binds very nicely to this DCC receptor, which is the known netrin receptor or main receptor. However, if you do immunoprecipitation in vivo, so here what we did, we collect hindbrain or spinal cord from mouse, here it's T2S spinal cord, and then we did immunoprecipitation of the ROBO3 receptor, and we blot with an antibody against netrin. So it's done in either wild type or mutant. And you can see that you get a band here that shows you that netrin is still in a complex with ROBO3. And it's specific because in the ROBO3 mutant, you lose this netrin partner. So it's not direct binding, but it seems to be close enough to ROBO3 to be immunoprecipited with ROBO3. So we move to this other receptor and see, so okay, what happens? Could it be the receptor involved in this system? So here I'm just showing you what are the netrin-1 receptors that are known so far, but I will just focus on this DCC receptor. So again, you take, if you take rhombic clip from the DCC mutant, put netrin in the floor plate, there is attraction of the neurons, of the pons. Now if you remove DCC, again, like ROBO3, you prevent the attraction. If you look at the phenotype of DCC mutant, it seems to be much more severe and more perturbed, but you kind of still see similar thing. Neurons, they will migrate to a point, some of them will leave the stream before, but they will never reach the midline as well. So it's not exactly like the netrin or the ROBO3, but there are also some defect, and the important thing that the neurons cannot make it to the midline. And in fact, what we found by doing co-immunoprecipitation studies in vitro and in vivo is that ROBO3 and DCC, they can interact together. So here you express cells, you try to co-IP. It's not slit dependent, you don't need slit, and slit doesn't modify the interaction between ROBO3 and DCC, which is not a surprise. It's also not modified by netrin. There is binding to ROBO3 independently of netrin or slit. That's in vitro, but also that's now in vivo. You take the DCC wild type, you can immunoprecipitate ROBO3 and DCC. You take a mutant, you lose the interaction, so it shows you it's specific. And the same, you take a ROBO3 mutant, you lose the interaction. You take the netrin knockout, you still have interaction. So ROBO3 and DCC can form a complex, and it's independent of netrin. Okay? So we went a bit further and even mapped what was the interaction domain between the two receptors, and we found that in fact the P3 domain, a cytoplasmic domain of DCC, is binding to this region here that covers the CC2 and CC3 domain of ROBO3. So that's where they interact. And what Pavel has done is to use a construct that is lacking this interaction domain and electroporate it in the mouse ROBO3. And I showed you before you could rescue normally some migration here when you electroporate the wild type ROBO3. But when you electroporate this mutant ROBO3 now that is not able to bind DCC, you don't restore the migration. So it seems that really it's important to have this interaction maybe with DCC to trigger this interaction. And we went a bit further and studied, okay, netrin doesn't bind to ROBO3, it binds to DCC, they seem to form a complex. Could netrin binding to a DCC or netrin receptor influence ROBO3 somehow? So what we did here was to take some cells, P19 cells, so they are mouse carcinoma cells, and those cells, when you differentiate them with retinic acid, they start expressing ROBO3 and DCC. They don't express it in basal condition. Okay, so you don't need to transfect DCC ROBO3, they just have them after retinic acid treatment. And in those cells, again, if you have no natrine, ROBO3 is not phosphorylated. Now, if you have natrine, you can have a phosphorylation of ROBO3 that is induced by natrine. And if you put PP2, which is an inhibitor of Src kinases, you can completely block this phosphorylation. So natrine binding to the cells induce ROBO3 phosphorylation, and this is prevented by Src kinase inhibitor. So what I want to show you today is what Eicher has done is also to transfect cells with overexpressing Src. You can also induce the phosphorylation. And if you overexpress a Src that is mutated in one amino acid that acts as a dominant negative, you don't trigger the phosphorylation. So it's really seems based also on other studies with other drugs that is really mediated by Src. And we also mapped what is the tyrosine that could be phosphorylated in the cytoplasmic domain. So there are only a few tyrosine in this domain. And by making some mutagenesis, we could find that the important tyrosine is this tyrosine 1019 that is around here, just close to the transmembrane domain and the first CC domain. And as you can see here, if you mutate this tyrosine and add natrine, then you don't have a very significant phosphorylation. And if you mutate this other neighboring tyrosine, 1002, then you still have the phosphorylation. And this tyrosine is conserved in ROBO3 receptors. You can see it here. And again, Pavel went to try to rescue the migration by expressing the ROBO3 that is just mutated in one amino acid with tyrosine. And if you do so, then you don't rescue the migration. So a single mutation at this tyrosine is enough to prevent the rescue. So it really shows you that this is relevant for attraction. So this is in the mouse. Then we went again to other species. So I told you SLIT doesn't bind to it, doesn't do anything, but you apply natrine, it does something to ROBO3 in the mouse. Is it also the case in other species? And the answer is no. Again, an example, this is mouse, you apply natrine, you induce phosphorylation. You take zebrafish, you don't induce any phosphorylation. So it's, again, this feature of natrine and this interaction of natrine's ability to activate ROBO3 is only in mammals, it's not in non-mammals. So what we think in mammals, so it doesn't mean it doesn't have another function, okay? It's possible that it does something to ROBO-repulsion, but we think the primary function, at least in many neurons in the hindbrain, is to act as a co-receptor for DCC or possibly other natrine receptors. And then when natrine will bind, it will activate ROBO3, and that will trigger attraction. If you take non-mammals, SLIT will bind to ROBO3, it will dephosphorylate, and then we don't know what it does. So we are a bit there now. So what ROBO3 then does in non-mammals. So just to show you, I don't think I have much time left, no? 5 minutes, that's good. So rather than coming this, is just some, we can talk about that later, just some thoughts about what it did in evolution and why we think it might be important and have helped shape the mammalian brain. But I mentioned the non-mammalian species, and I just want to show you that now we have tools that will allow us to study this, and just to show you some preliminary results on other things to try to trigger some discussion. So we've been working with Dietmar Schmucker in Leuven, and Dietmar has found a way to use talent and CRISPRs in Xenopus. So he's using Xenopus tropicalis, and as you can see here, they are nice, they swim. And if you do mutation of ROBO3, this is what you get. They have this curly tail, and they are swimming like this. So surprisingly, they survive. So we were hoping that they will go through metamorphosis. It's still ongoing. And we also hope that it will go to germline. But it seems here again that if you perturb ROBO3 in Xenopus, you do something that is not good for the frog. So it's really something that we want to study because, as I mentioned before, the frog is also interesting because it goes through metamorphosis. And there's some rearrangement of the circuits during metamorphosis. And we like to see if ROBO3 is important before metamorphosis, but also after metamorphosis. So that's the frog. So now we have a system where we can test ROBO3 function. But there's also the fish. In the fish, there's a ROBO3 fish mutant called Twitch twice. And there's also now recently, Michael Granato generated also a DCC fish. So I just want to show you some preliminary results obtained by Jörn Schweitzer in Freiburg, with whom we've collaborated about this. So this is a double heterozygous Wolf3 DCC. You can see many commissures here. This is the hindbrain, the trigeminal ganglion is here, and it's stained with a general marker acetylated tubulin. And what is surprising, if you take the DCC mutant, you can see that many commissures are left. I mean, it's very hard to see any difference between a fish that has DCC and a fish that has no DCC. So it's a bit weird, because then what DCC does? Now you take the Wobo3 fish mutant, so it lacks some commissures, but you can see that many commissures are also still present in the Wobo3 fish. But now if you make a double mutant, it's like a commissureless fish. It has no more commissures. So it shows you that you don't need to have 20 receptors, and those two, they are really worth studying, because the two together, also in the fish, they seem to control the development of all the commissures. So now we are trying to see, by making rescue experiment, other kind of experiment, how does it work? Do they interact together as well, or is it through another receptor? Okay. Here you can see that maybe you lose the attraction, and Wobo3 is not able to prevent Wobo repulsion. So you have two things: axons are repaired, and they're not protected anymore, so they cannot cross. So it's all the kind of model that we are testing. So just to conclude, 2 slides to show you some recent results, and to show you that there are still open questions, interesting questions to answer about this Wobo3 and its role in evolution. So we've been interested more recently in another system that is called the habenula. So the habenula has 2 main nuclei called the lateral habenula and the medial habenula, and it's a relay system between the forebrain and the midbrain and hindbrain. It's located dorsally in the epithalamus, so in the diencephalon, and it receives input from the diagonal brain of Broca, also from the septum, and through a tract that is called the stria medullaris. Okay? And then it projects, so the medial habenula will project to a nucleus called the interpeduncular nucleus, and the lateral habenula will project to the, mostly to the substantia nigra. Okay? And in the fish, it also exists, like in all vertebrates. And in the fish, it's interesting because this nucleus is completely asymmetric. The medial habenula, the lateral habenula, they don't have the same size on both sides. And also the type of axon that they project to the interpeduncular nucleus, the morphology of the axons is also different between the right side and the left side. And what is interesting with this system for us in terms of commissure is that you can see if this is in the fish, one axon that was traced, when it reaches the midline, it crosses it several times. Okay, it seems that unlike all the other axons that only cross only once, in this system they go around and they like a robo mutant by themselves, they cross the midline several times, which is also something interesting. What is the mechanism then that is controlling guidance in the system, and why do they cross several times? And there's not much about the molecule that control the development of the habenula, and there's just a little bit known that they respond to netrin if you make expunts, but the mutant was not analyzed. And also they tend to grow here because they are repaired from the tectum by semaphorins, but that's it. So when we were doing our studies about Wobo3, I mentioned its hindbrain spinal cord, we were surprised that when we did staining of an embryo with Wobo3 at E16, which is quite late, and normally there's not much Wobo3 expressed anymore, you can see here that you have a very nice staining of the medial habenula, the tract, all the way to the interpeduncular nucleus. So nothing else is labeled now in the brain with Wobo3 except this tract. And this tract, it's a bit weird because it reached the midline well before, around E12, and still at E16 they express Wobo3, P0 they express Wobo3, and in the adult they even express Wobo3. So in this system they express it very nicely, but it's not downregulated, and it's the only system rostral to the hindbrain where you have Wobo3 expression. So to study this tract more nicely, we've been working for some time now. So in the lab, it's in the Institut, it's David Godefroy and Morgan Bell, and it's in the lab who have been working on it. We've been working on embryo clearing. So for those of you who don't know, so the fish, always people say it's nice, it's transparent, the frog is transparent, the mouse is not transparent. It's true, it was true, but now you can make it transparent by using some tricks. And by also playing on condition, you can now see the tracks inside the brain in 3 dimensions. And it's very useful for this tract. So again, you have the medial habenula, the fasciculus retroflexus is the name of the connection of the medial habenula to the interpeduncular nucleus. And here you have the pons morcodalis, so you have to think that the whole brain is here, anterior, posterior, ventral. So now if you make this 3D images, and that's called, it's with light sheet imaging, you can see all the projection, the left side, the right side. You can follow the axon through the brain all the way to the hindbrain, and then you will see where they connect to the interpeduncular nucleus, the shape of the axons here, and you can really move inside the brain to follow the axons. You can follow single axon, that's at E16, but it works also at least at P0. And we are working for other stages. And you will see that you can see all the connections when they reach it. And in the mass as well, you can see that it seems that the axons, when they reach it, they seems to cross several times the midline as well. It has this shape here, like an 8 or an S when they reach the midline. So this is just, again, Robo 3. And we are starting to screen for many mutants to try to find genes and receptors that control this choice. I'm just showing you two striking examples, which are NETRIN1 and DCC. So you can see in the NETRIN1 mutant and the DCC mutant, some of them make it, but most of them are not able to reach the midline. And also what is interesting, you have many axons now that we cross dorsally. Some of them will go to dorsal, they will cross the midline here. In the DCC mutant, many of the axons, they will go rostrally and not caudally anymore. And you see that at the midline, some of them still make it, but they have problem crossing. So this tool, again, is very useful to study rapidly many mutants. And the last slide is to show you what you have in the Robo3 mutant. And then the axons, they still make it to the floor plate, so which is not the case in other regions of the brain. But they have some problems, and they seem to stay here and to stay along the floor plate, as if they were not repaired. So it's a bit surprising, because in this system, Robo3 is maintained, there's a phenotype, but it doesn't really fit with everything I've told you before. So we don't know what's happening here, and clearly it suggests that in different neurons, those receptors, based on other co-receptors, they might have completely different function. Okay, so we stop here, and just, of course, it's a work of many people in the lab. I mentioned all of them during the talk. I think I didn't forget anything. No, Chloe, who has been working with David to develop the ultramicroscopy a lot. And I think I will just stop here. I mentioned all the collaborators in the talk. Thanks a lot for your attention.